Hello, everyone. My name is Lian Pan. I'm a Senior Marketing Manager at GenScript. Today, I invited Dr. Liu Song-Ying, who is the Group Leader of Antibody Discovery at our Antibody Production Department, to talk about the challenges and solutions on antibody drug development. Dr. Ying received his PhD from University of Massachusetts Medical School, specialized in antigen presentation and CD4 T-cell epitope selection. He then joined Pfizer as a postdoctoral fellow, specialized in immunogenicity of the therapeutic biologics. Dr. Ying has extensive experience in antibody drug development, especially in DNA immunization, high-throughput FACS screening, ELISA kit development, and anti-idiotype antibody development for antibody drug PD/PK studies. In this webinar, he will first give an overview of the antibody drug development and then talk about the antibody drug development platforms and new technologies. After that, he will touch base on anti-idiotype antibody development, followed by some key studies on the antibody drug discovery, which will wrap up Dr. Ying's presentation. Then I will spend a few minutes to introduce you the antibody services at GenScript. Hope you can take this opportunity to learn from our experts and ask questions that you may have regarding antibody drug development. To ensure the sound quality, all the attendees are muted. Please feel free to type in your questions in the chat box in the lower right corner of your screen throughout the talk. Dr. Ying will address your questions together after the service introduction by me. Without further ado, let's welcome Dr. Ying give his talk on the challenges and solutions. Dr. Ying, welcome.
Thank you, Lian, for the introduction, and thanks everyone for attending this webinar. As you all know, that antibody drug market is booming, with annual revenue over $100 billion. In a recent review, among the 20 top-selling biopharmaceutical products in 2003, half of them are antibody drugs, including the top three. Notably, as you can see that only Humira, an anti-TNF antibody, achieved $11 billion in 2013. Antibody drug development process is complicated, and here I list several essential steps. The process starts with target selection based on literature reports or in-house tests. We then prepare the antigen for immunizations and Hybridoma generation if using Hybridoma approach, which I will discuss more later. After you obtain hundreds of thousands of hits from screening, you can further narrow down the list by performing functional assays for selection of leads. Lead optimization is usually followed, including chimeric antibody generation, antibody immunization, and antibody affinity maturation. After that, large scale of antibody production will be performed for further toxicity and efficacy studies, as well as other characterizations such as stability and developability studies. There are several technologies to generate therapeutic antibodies. Hybridoma approach with human transgenic mice or B cells from immunized human body can directly generate human antibodies. The most popular one is still Hybridoma approach with rodent system, in which you get rodent antibodies and then perform humanization to get humanized antibodies. Besides the Hybridoma technology, there are other technologies for generating therapeutic antibodies, including the SLAN technology, which recovers the variable region sequence from specific single B cell from immunized animals and is able to generate humanized antibodies. Phage or yeast display is also an option to get humanized or human antibodies depending on the library you build. More recently, next generation of antibody sequencing from naive or immunized individuals can also be applied to generate human or humanized antibodies. Here are several approved antibody drugs from each technology platform. As you can see that most of them are derived from Hybridoma approach as fully human antibodies from transgenic mice as colored in red, or humanized antibodies from rodent systems as colored in blue. And as you can see that the Hybridoma approach is still most popular one. As mentioned, Hybridoma technology is traditional since 1970s, but still powerful. By 2015, FDA has approved 44 therapeutic antibody drugs, and 41 of them were derived from Hybridoma technology, and only 3 from phage display platform. The Hybridoma technology still generates high market value by inputting more therapeutic antibodies into the market, as you can see from the table below. Here are the critical paths for antibody lead identification by Hybridoma approach. We start with regional preparation, including immunogens, positive screening and negative screening materials, as well as assay reagents. Animal immunization is followed, and meanwhile, assays for anti-serum titer test, Hybridoma screening, and his characterization are being developed. The next step is to select animals for fusion based on titer test or other functional evaluation of the anti-serum from each immunized animal. An average of 10,000 to 20,000 Hybridomas can be obtained, which will be subjected to several rounds of binding, ligand blocking, and functional screening. After multiple hits are generated, subcloning is performed to achieve single stable monoclone for lead selection based on further biophysical and functional assessment. Lead optimization, such as affinity maturation and humanization, is often performed. Next, the lead will be subjected to toxicity and efficacy evaluation in animal models for candidate selection. These are the critical paths for antibody lead identification by Hybridoma approach. Conventional Hybridoma generation procedures follow immunogen preparation, animal immunization, fusion and Hybridoma screening, subcloning and Hybridoma cryopreservation, and antibody production. GenScript has extensive expertise in all of these critical paths, from multiple approaches on immunogen preparation to a variety of choice on host animals. We also have high-efficient electrofusion platforms and high-throughput screening platforms for identifying binders against the most native epitopes. Moreover, we have a very experienced team for project management, and in the last 10 years, we have successfully delivered more than 10,000 antibody projects, including over 200 therapeutic antibody projects. In the next couple slides, I will expand a little bit on each of our expertise in the critical paths. We have multiple choice of immunogens on a target basis, from peptide, which can be native or modified, to protein, including secreted proteins, extracellular domain or membrane proteins, and membrane preps. Whole cell and DNA immunization are suitable for receptors, including GPCRs, surface antigens, including immune checkpoint proteins, ion channels, and transporters. Virus-like particle is efficient in incorporating membrane targets for immunization. We can also use a combination of the various immunogen options following specific project requirements. So these are the multiple choice of immunogens that we have. In the case that our human target has high homology with the rodent counterparts, we have the proprietary ImmunoPlus technology to break immune tolerance. In the case shown below, before applying this technology, all the 4 mice did not generate sufficient antibody response against the high homology target. However, after applying the ImmunoPlus technology, all mice generated significant anti-serum titers. Another application of this ImmunoPlus technology is to develop surrogate antibodies such as mouse anti-mouse antibody for target validation. This is very useful and it's quite common recently. Next, I will talk about our specialized service, gene immunization or DNA immunization. Gene immunization is a powerful method to generate mAbs against membrane-bound proteins, such as immune checkpoint proteins, for more native conformation, and GPCRs and ion channel proteins that are hard to express, purify, and prepare as immunogens. In this method, host animals are injected with DNA plasmid encoding target proteins to elicit immune response. In the gene immunization option, immunogens can be DNA plasmid only, or DNA priming followed by boost immunization of cells overexpressing target protein or extracellular domain of target protein. Multiple mouse strains, including gamma transgenic mice for high affinity monoclonal antibodies, and rat can be selected for host animals. The plasmid are administered through gene-gun-mediated DNA delivery at 3 to 50 microgram DNA per animal, with or without 5 times 10 to the 6 overexpressing cells or 10 microgram protein boost. After the immunogens have been prepared, we could start immunization and hybridoma fusion. As mentioned, multiple choice of animal strains are available, and in most cases, they can reach a very high titer after third immunization with an average titer of 512 K. The electrofusion method used guarantees high cell fusion efficiency to increase the size of library. We observed an average fusion efficiency of 1 hybridoma over 5,000 B cells, resulting 10,000 to 15,000 hybridomas per animal. After we obtain a large number of hybridomas, the next step is to perform hybridoma screening for binders. We have multiple nature epitope-based high-throughput screening platforms. For soluble target, both capture and indirect ELISA are in place. For membrane target, we have BD fax caliber with high-throughput loader and high-throughput IQ screening system, which can read a 96 well plate in 3 to 5 minutes by fax. We are also equipped with the homogeneous high-throughput match binding assay system for membrane target binding screening. With all these high-throughput platforms, we are able to identify numerous hits of binders for therapeutic antibody development. After we get a large number of hits, multiple characterization methods will be conducted for lead selection, including the epitope binning to see whether they bind to the same epitope or not, and a relative affinity ranking for selecting high affinity clones. Definitive affinity measurement can be performed by Biocor, and IC50 or EC50 can be determined in cell-based functional assays. Target specificity and cross-species reactivity can also be evaluated for lead selection. Here we just show some examples of those epitope binning by ELISA and by Octet. Next, I will talk about this is a quick summary for advantage of GenScript's antibody drug discovery platform, including multiple immunogen and host animal choice, an optimized adjuvant and immunization schedule, as well as our ImmunoPlus technology to break immune tolerance for high homology targets and for surrogate antibody development. Our high-throughput platforms enable us to efficiently screen numerous hybridomas and obtain multiple hits. In vitro and in vivo functional assays are in place for lead selection. Downstream lead optimization, such as humanization and affinity maturation, as well as large-scale antibody production, are also our expertise. Using those platforms, we experienced many successful cases for soluble targets as well as membrane proteins, such as single transmembrane immune checkpoint proteins and multiple transmembrane GPCRs and ion channels, using ECD proteins, membrane preps, recombinant cells, plasmid DNAs along or their combination. We have a lot of experience in generating therapeutic antibodies against all of these targets shown here. In the next few slides, I will briefly talk about anti-idiotype antibodies, which is a must-have tool for antibody drug development. Anti-idiotype antibodies are antibodies against the variable region of antibody drugs, with the antigen binding specificity defined by the distinguished sequence in the V region. Here is an example showing that anti-idiotype antibody against a human IgG antibody drug only recognizes antibody drug, but not human IgG control. There are three different types of anti-idiotypes. The first one is antigen blocking or paratope specific, which inhibits the antigen binding when bound to the antibody drug and can only detect free antibody drug. The second one is non-blocking or non-paratope specific, which does not inhibit antigen binding and can detect the total antibody drug, including free, partially bound, and fully bound. The third one is not that common, which is complex specific and can only detect bound drug. In most of anti-idiotype projects, we will generate the first two types, which are antigen blocking and non-blocking, because those are most useful in their applications. There are two major applications for anti-idiotypes. It can be used to measure the antibody drug level in patient samples in pharmacokinetic assays. It can also be used as a positive control or reference standard for anti-drug antibody in immunogenicity assays. I will expand a little bit for these two applications. There are four typical PK assay formats. The first one is to use antigen to capture the antibody drug in patient samples, and then detect with the secondary antibody. All the other three formats involve using anti-idiotype to capture and detect antibody drug in the samples in sandwich or binding assays. And you can see that in the second format, you can code the anti-idiotype antibody on the plate, then which can capture the antibody drug in the human serum, and then you can detect with a labeled form of the anti-idiotype. In this case, you can quantify the antibody drug in the samples, and the other several formats following basically the same principle, just have different detection or capturing reagents. I will switch the gear to immunogenicity. Immunogenicity of therapeutic antibodies is caused by various product-related and patient-related risk factors, and it can cause different therapeutic outcomes in terms of drug efficacy and patient safety. Various methods have been established for measuring and predicting immunogenicity, and FDA has guidelines for risk-based mitigation efforts for immunogenicity. Right now, immunogenicity assessment is a must-have evaluation before approving the antibody drug. As mentioned, there are various assay formats for measuring immunogenicity, and each has its own advantage and disadvantage in regard to sensitivity, specificity, and drug interference, and the method can be selected on a project-specific basis. However, regardless of the method selected, positive control or reference antibody is needed for appropriate immunogenicity measurement. Anti-idiotype antibodies could serve this purpose. Purified anti-idiotype antibodies can be spiked into the matrix selected for screening assay and used as a reference standard. In this case, you can quantify the anti-drug antibodies in the samples using this reference. And you also need a negative control, which can be a pool of sera from several non-exposed individuals. Here is an overview of the immunogenicity of several FDA-approved antibody drugs. In general, the murine antibody has much higher immunogenicity rate compared with humanized or fully human antibody drugs, as the label in the red, which are murine antibodies, has a immunogenicity rate usually over 80%, and the humanized or fully human antibodies such as Humira has a much lower immunogenicity. Regardless, almost all of the antibody drugs have more or less immunogenicity, and they can cause different therapeutic clinical outcomes. That's why immunogenicity evaluation is a must-have. Next, I will give a couple representative case studies of antibody drug development. In the first case study, the target is a soluble 20 kD cytokine antigen, and the core requirement is to develop functional mAbs recognizing target in circulation. Using the protein as immunogen and our native select capture ELISA, we were able to identify multiple high-affinity hits with KD in the sub-nanomolar range, which are currently in downstream lead optimization. In our experience with protein immunization, the titer is extremely high, usually over 512 K at 1 to 8,000 dilution of the anti-serum. And we can have several hundred and thousand binders from several rounds of binder screening. In the second case study, the target is a membrane-bound receptor, EGFR. We used overexpressing cell line as the immunogen and a high-throughput platform for screening fax binders. We have several fax binders in the end. The clone 5E10E9 has a high affinity also in the sub-nanomolar range and is very efficient in inhibiting EGF-induced carcinoma cell line proliferation as approved in the functional assay. The data are shown here. The third case study is to use DNA immunization followed with overexpressing cell line boost for multi-transmembrane GPCR target GLPIR. We successfully identified several fax binders using GenScript derived GLPIR stable cell line. In this case study, we use the DNA immunization to prime the animal, then use transient overexpressing cell line to boost. And in this case, the titer is much higher, and we successfully have several binders, and those can be very useful in following downstream application. Among the fax binders in the DNA immunization case study mentioned earlier, the clone 1F1E7 can also efficiently detect GLPIR incorporated in virus-like particle by Western blot, as you can see from the figure in the left panel. The last case study I want to share is for anti-idiotype antibody. Since 2009, GenScript has been specialized in making anti-idiotype antibodies to support our customers' PK and PD and immunogenicity evaluations of antibody drugs. Our record of success is 100% with more than 100 projects. The left panel shows anti-idiotypes with high affinity with KDs in sub-nanomolar range, and the right panel shows anti-idiotypes with high specificity that only recognize antibody drugs but not isotype control and total IgG in human serum. In all these anti-idiotype antibody projects, we have delivered more than 10 clones for each antibody drug, and then the client usually asks us to do some kind of pairing studies for the PK or derive a pool of monoclonal antibodies or polyclonal antibodies as a reference for their immunogenicity studies. With that, I board to Lian for general antibody service at GenScript, and I'm more than happy to answer your questions right after Lian's service introduction. So Lian, please.
Thank you. I really, appreciate that Dr. Yin gave such an informative talk on antibody drug development. I surely learned a lot. Also like to thank you all for attending this webinar. I will spend the next few minutes to quickly go through our antibody services to help you understand how GenScript can help you in your antibody drug development process. Meanwhile, you can type your questions in the chat box at the lower right corner of the screen. Dr. Yin and I will address your questions all together right after my service introduction. So this is an overview of the services we offer at GenScript. Besides the antibody drug discovery related services, which include a therapeutic antibody discovery platform, anti-drug antibody, surrogate antibody, and immunogenicity assays, We also offer reagent antibody services for polyclonal antibody, monoclonal antibody, phosphor-specific antibodies, neutralizing and blocking antibodies, as well as antibody pairing. We also offer catalog antibodies, diagnostic antibodies, and immunoassay development. GenScript has over 12 years experience in antibody production. Our animal facility is ALAC and OLAW accredited, hosting over 8,000 rabbits and 12,000 rodents. We are equipped with high production capacity. We can produce 500 to 600 polyclonal antibody and 50 to 80 monoclonal antibody each month. If you have any antibody production need, GenScript is the one-stop shopping place for you. We offer a variety of antibody production services. Depending on your applications, you can choose either antibody drug development related services. For example, we offer anti-idiotype antibody to facilitate your antibody drug PK and immunogenicity studies to fill the FDA requirement. If your targeted proteins are membrane proteins, ion channels, and immune checkpoint proteins, we offer DNA immunization services to circumvent the requirement for in vitro protein antigen production. It's a very powerful tool for antibody production against problematic antigens. If you need recombinant antibody at the later stage of antibody drug development, and we offer high-throughput gene-to-antibody services, guaranteed mammalian expression of full-length antibodies and also antibody fragments, as well as gram-level stable cell line services. For antibodies used for applications such as vaccine blot, IHC, flow cytometry, IP, chip, etc., you can choose our MonoExpress or PolyExpress services for your custom antibody development. We offer the fastest turnaround time with application guarantees. In the next 3 slides, I will go over some details of our specialty services, including DNA immunization, anti-idiotype antibody, and MonoExpress monoclonal antibody production. Let's first take a look at the DNA immunization service details. The project usually starts with gene synthesis and plasmid preparation. Before animal immunization, the plasmid will be transfected into cells for expression validation. This step typically takes 2 to 3 weeks. After animal immunization by gene gun, followed by several rounds of boosting, which typically takes about 8 to 10 weeks, the cell fusion and screening, subcloning, single clone expansion, and cell preservation will be carried out sequentially. On your project need, we can also deliver you the purified antibody at the amount that you specified. For anti-idiotype antibody production, we offer both polyclonal antibody and monoclonal antibody services. The starting material, QC standards, and deliverables are specified in the table here. Our production scientists have extensive experience in anti-idiotype antibody development, and our success rate is 100%. If you have anti-idiotype antibody projects, I highly recommend that you seek our expertise to ensure the high specificity and affinity of your desired anti-idiotype antibody. We can also help you to develop the downstream PD, PK, and immunogenicity assays. If you are looking for antibodies that work for your own applications, I recommend you to go with our MonoExpress premium package. This premium package includes antigen production. You just need to provide us the target protein sequence. Our production scientists will take care of the rest. We will deliver you 10 ELISA positive supernatant for you to evaluate in your own application setup, and pick up to 5 hybridoma cell lines for the final delivery. We guarantee that at least one clone will work for your application. In the final delivery, we also include 2 mg purified antibody and 200 microgram recombinant protein, which is a big plus for some customers who also need recombinant protein for their projects. To summarize our key services, for antibody drug development, I would suggest you to choose DNA immunization services, which offer a solution to antibody production against problematic antigens. Premium hybridoma services, which ensure you a successful start for your therapeutic antibody development. Recombinant antibody production services, which offers flexible production scale from microgram to gram level, and anti-idiotype antibody services, which have a proven success track record of 100% success rate. For reagent antibody production, I strongly suggest you to choose the MonoExpress premium package for your monoclonal antibody production and the PolyExpress premium package for your polyclonal antibody production. Both packages offer application guarantees. Besides antibody services, we offer a broad range of services to make your research easy. This slide gives you a general overview of the services we offer at GenScript, including gene synthesis, subcloning, peptide synthesis, protein expression and purification, custom antibody production, discovery biology services, and single cell lines, all with the goal to make research easy. More than 11,000 peer-reviewed journal articles have cited GenScript services and products, making GenScript the most frequently cited biology CRO in the world. I'm very proud to be part of it. So this wraps up my introduction on our antibody services. So now it comes to the Q&A section, and let's take a look at the first questions. Let's go. So the first question here is, how is antibody drug development different from regular antibody development? Dr. Jin, could you comment on this?
Yeah, that's a great question. Because I was asked this kind of question again and again. So for general reagent antibody development, compared with antibody drug development, the QC is much simpler and the process is much more streamlined. For example, in a general, you won't have a reagent antibody for ELISA usage. Then we just have to do the simple immunization and fusion and then hybridoma screening, all based on ELISA, and do some subcloning and antibody production, all based on ELISA. And we don't have to be exhausted because we only need several binders, and we just won't deliver 5, that's enough, because 5 antibodies should be enough for your usage as for ELISA basis. On the other hand, the therapeutic antibody development is totally different because there are many more QC in each step. From the beginning, you have to worry about which immunogen we should use and which animal strain we should use. And there are multiple QC besides ELISA, such as FACS, such as functional assays, such as those epitope affinity ranking, because the functional hit rate is much lower than the regular ELISA binder. So you have to make sure that you start with a lot of hybridomas and have multiple hits, then you will be able to get several leads. So to summarize, the key difference is the process is different based on your application. Your reagent antibody, you just need a reagent, just need its binding in ELISA or WB or FACS. For the therapeutic, it must have a function that can behave, and it can behave well in vitro and in vivo. So that's a key difference.
Thank you, Dr. Ying. I saw the second question is for DNA immunization, what's your success rate?
So for DNA immunization, currently we have about more than 50 projects accomplished and about 20 ongoing. So for single transmembrane proteins such as immune checkpoint family members, the success rate is over 95%. For multiple transmembrane proteins such as GPCRs and ion channels, the success rate is lower. It's about 50 to 70% due to the complexity of the target itself and due to the different conformation, and especially the native conformation of the targets.
Thank you. There is another question regarding anti-IDs antibody generation. So it's the anti-idiotype antibody generation. And the question is, do you use the full-length antibody as the immunogen?
Yeah, that's a good question, because I think you ask this question because you worry about the antigenicity of the Fc, because the anti-idiotype antibodies are only against the V region. So in general, for most of the cases that we accomplished or is ongoing, we use the pepsin digested FAB2 as the immunogen to increase or to enhance the antigenicity of the V region. However, in certain cases that it's hard to digest the antibody drug to get FAB2, we do use full-length antibody, and we still get success in identifying specific antibodies, recognizing antibody drug, but not recognizing the S-type control or human serum. So in that, even though the hit rate is a little bit lower, that usually you get more than 20 or 50 specific binders in using the FAB2 as the immunogen. In using the full-length antibody, you have usually less than 50.
That's great information. Thank you, Dr. Ying. So we have another question here. What is the general timeline for antibody drug development?
So as mentioned, the antibody drug development process is complicated, and for the binder screening, usually takes about 6 months to get yearly FACS binders. For more in vitro and in vivo functional assays, it takes about another 6 months. So you will add to 1 year to get a functional lead. And if you want to do humanization and the following lead optimization, it takes about another 6 months. So total, you will have a humanized antibody within about 1.5 years, with 6 months for the binder and 6 months for the functional assays for lead selection, and another 6 months for the lead optimization.
Wow, that's a long developing time from the target selection to the humanized antibody. I think we have the last question here. The question was asking, do you have the transgenic mice? I'm not quite sure which transgenic mice they are mentioning to. Maybe the humanized transgenic mice, do we have?
Yeah, I think so. I think it's asking whether we have, because I mentioned using the transgenic mice, you can directly get a fully humanized antibodies. And yes, we do have the service right now because we are the preferred providers for transgenic mice, which is the second generation transgenic mice provider. We are the preferred provider, and we can provide those service together with the company that have the transgenic mice.
All right, I think that's all for the webinar. I want to thank everybody for attending this webinar and representing GenScript. We wish you the great success in your research. Thank you. Bye bye.